don't need to wait in the lobby. So they can bypass and straight away get into it, whoever has the link, right? So what was the take home message on this slide? Anybody? Um, Here we talked about ribose and deoxyribose. Deoxyribose in DNA and ribose in RNA, right? The difference is that at 2' carbon position number 2, we call it as 2' position, and prime I indicated that is the difference between those carbons that are there in these sugar molecules versus the carbons which are there in nitrogenous bases. They are numbered as 1, 2, 3, 4, 5, 6, right? And here these carbons are numbered as 1', 2', 3', 4', 5', right? So these are your uh, the carbons which are belonging to these sugar molecules, ribose in case of DNA, sorry, RNA, and deoxyribose in case of DNA. Why deoxyribose? Because at position number 2 on ribose there is O here, and in deoxyribose the O is removed. So there is just H here. So that O is removed. So that's why we call it as deoxygenated, deoxyribose, right? On position number 3, there is OH here. On position number 4, why it is important? Because with one it binds through that oxygen molecule, and that gives this molecule the five-cornered shape, right? A pentagon shape, five corners. Because the fifth carbon is not in the main pentagon or the main ring that is on the side, but that fifth carbon is also important because phosphate group attaches here. So that was the take-home message from this. Here on this slide, we talked about the purines, adenine, and guanine, double ringed nitrogenous molecules, nitrogenous spaces, we call them as, right? And pyrimidines, which are the single ringed. Any questions? Right, so I'm trying to join बस वो क्लास चल रही क्लास ऑनलाइन में जा रहे हैं आप क्या क्लास की है नीचे से मैं तो उसको जल्दी करना है मैंने जाना पर ठीक है पर जाओ ना मैं अरे वो चार्जर हो यार चाहिए आ गया चार्जर आपका अभी नहीं आया सुबह ले लीजिएगा क्या करना okay so we have that cytosine thymine and uracil thymine and uracil replace each other. In case of RNA, there is uracil, and in case of DNA, there is thymine. So that's the distinction between these two, right? If you look into their molecules, that is, this uracil is basically it does not have that methyl group. So thymine is, we can say, methylated form of uracil, right? When I say that, yes, it is. It seems like so. If you remove that methyl group, it becomes uracil, right? And this way there is that sound coming in. So Madam Fozia, आपका mic on है. Right. So that's what it is. And those nitrogenous bases, when they are put on those sugar molecules, we talked about that yesterday. It becomes a nucleoside. So this adenine becomes adenosine, and you put a phosphate group, it becomes adenosine monophosphate. That shape, it's called as nucleotide. And we also explored that those nucleotides are actually, when they are formed, so those free nucleotides are not in monophosphate shape, they are in triphosphate shape. And when that nucleotide is incorporated into a growing chain of DNA, those two phosphates, they run away, right? And that property has been utilized by one of the company for sequencing. What was the name of that company that uses these two phosphates? What these two phosphates are called? They are called as pyrophosphate.
Pyrophosphates.
Right? So what was the company that
Pyrophosphate.
that company is Roche, right? And their method
Raw.
got famous. It got famous as pyrosequencing, right? And there was another name for that, 454. 454 was the project of Roche, the project number of Roche. Number 454, that was given the task to work a research or R&D for these sequencers, right? To come up with that sequencing methods and sequencing machines, we call them as sequencers. This is just a revision of Watson and Crick double standard DNA model. And according to that, what we see over here is that DNA strands, they get the directionality such that this on one side that 5 prime end is free because on 5 prime end you see a phosphate. On other side there is an OH group that is free, so that is 5 prime end and that is 3 prime end. And this strand is upside down. The other one is pointed upwards. So one is upside down, one is pointed upwards. So they run in anti-parallel fashion such that this guy goes like this: phosphate sugar, phosphate sugar, phosphate sugar. In the end, there is an OH group. Again, on this side, phosphate sugar, and ending up on this side exposing the arms, and on this side exposing the feet, right? So that is the feet, OH, and that is the arm. So that is the head, that is the tail. So that is the direction, right? So 5 prime end to 3 prime end, and it grows from 5 to 3, right? And then here, this guy has 5 prime end on this side and 3 prime end on that side. So this arrangement actually, gives them the complementarity, right? So A attaches with T on the other strand, and C goes with G. So we know, we explored, we discovered that there are triple bonds. There are double bonds between A and T's, right? So if you have one side of the strand, if you make it single stranded, you can predict or you can make a synthesize the mic. I'm sorry, Rakib, your mic is disturbing. Uh, Somehow it switches on. I can mute all of you. There is this possibility. Audio settings. Settings. No, that's for me. That's for my own audio systems. Audio settings.
सर आपका माइक म्यूट हुआ हुआ है
So thank you so much for letting me know. Uh, okay, right. So somehow I got muted. So we have the template strands here. This phenomena was there, the DNA replication. So people explored that, and from where they came up with the idea for sequencing. And this method is called as sequencing by synthesis, right? Sequencing by synthesis, SBS. So what happens in SBS is you synthesize a new strand, just like it happens in the natural process replication. That replication is then replicated here and it is copied here, right? So we get the template, we have the polymerase enzyme. So there was multiple teams working on polymerases, multiple teams working on template preps, right? And combined together, all those things contributed towards sequencing. So finally, that sequencing was presented by this sequencing by synthesis thing presented by Fred Sanger, right? So that's why it got famous as Sanger's method. Other methods were also there, but this Sanger got the most successful, you know, utilization in life sciences community, right? So we need to have multiple things. We need to have template. We need to have polymerases. Other enzymes are also there. I'm not mentioning. So polymerase is the key one, the big one. So that's why I'm mentioning that. And then you need to have the runs of some nucleotides that should be already attached to the template. The thing is called as primer. So that white part is primer here, and then those yellow parts are your nucleotides that are coming in and attaching here. And suppose we are here at this point. So what you have here in the template is G. So the on the opposite strand there should be a C that should be incorporated, right? But before that, this previous G must have this OH free and available. So then this new nucleotide will come in, and that new nucleotide will come in the triphosphate shape, right? So then these two phosphates they are cleaved from here and they are broken, and then these two phosphates are called as pyrophosphates. So one of those sequencing platforms uh, uses these two pyrophosphates, as you already mentioned, pyro sequencing, right? Done by Roche. The company's name is Roche. But the sequencing method was using those pyrophosphate molecules. So that's why this got famous as pyro sequencing. So Roche was the company. Method got famous as pyro sequencing, and this was also it got famous as 454. That was the number of the project. So Raj 454 or pyro sequencing is the same thing. Okay. Roche is the name of the company. The method of sequencing is pyro sequencing. Okay. All right. So the other companies like Illumina that came up with the similar idea what original Sanger sequencing had. And there is one more company we checked yesterday. That was the Oin torrent. So in Oin torrent, we also know that on one side there is that pyrophosphate is going, but on the other side, the hydrogen ions are also coming out, right, in the mixture. These processes, these hydrogen ions as well as the water decomposition and condensation keeps on happening into that mixture, right? So that thing is going on. So that is generating the hydrogen ions in the medium. So that is changing the pH of the system. So that change in pH is detected by one of the companies called as ion torrent. So they are capturing the hydrogen ion, and that ion changes the pH. So change in pH is detected whenever a nucleotide is incorporated. There is a change in pH, right? So that company relies on that. That company got famous as ion torrent. Thermo Fisher Scientific adopted that ion torrent technology, right? Whereas the original Sanger method has these nucleotides. So if that nucleotide has OH, fine, this thing will attach. But if it has H here at position number 3, if you suffocate him, if you take its oxygen out, then it becomes dideoxyribonucleotide. Why dideoxy? Because on position number 2, O was gone. So this is deoxy. Then on position number 3, again that oxygen is gone. So that is twice or double deoxy. So dideoxy, right? So two times that thing is happening both on position number 2 and position number 3. So what is the consequence of taking out oxygen from position number 3? That new nucleotide will not attach here. So that will result in setting up this arrangement for this first generation of sequencing. So here we set up 4 reactions. In one of the tubes, we have everything good, except we use a combination for ATP, so normal ATP plus dideoxy ATP. So what's going to happen? We have the template, we have the primers, we have the polymerase, whatever we need, rest of the things. We had every salt and pepper here. Do that experiment, but except we get a combination or a combo for ATP, and we have a normal ATP and dideoxy ATP. The next tube has normal CTP plus dideoxy CTP, so combination for cytosine triphosphate. And the next one has for guanosine. The next one has for thymidine, right? So TTP, GTP normal plus plus dideoxy one. So we have the combinations for them. So based upon those combinations, we label those reactions or tubes as A tube and C tube and G tube and T tube, because rest of the things are the same. So where that difference is coming only from that particular nucleotide. Okay. And then you generate differential fragments here. So when this start synthesis of the new strand, what happens over here is that, for example, this is the primer, these two bases here, GGs, attached to CCs, and we are not seeing what we had in back of it. We are just starting from here, somewhere in the middle, just to make our point. Normally, that primer must, I mean, you should be wondering that it should not be two bases long. This is just for concept building, right? So that's right. The primer cannot be just two bases, but there is something which we had over there, but it's not that relevant to talk about that, right? So here we are starting. So that is the nucleotide that needs to be incorporated. The first one after this primer thing, that is a C should be incorporated because on the opposite strand, on template strand, we have G. So obviously in this tube, there will be a reaction. And again, there will be two possibilities. A normal C attaches or a dideoxy C attaches. If a normal attaches, it will continue. Here it continues because second time was also a normal C that was incorporated. Here at this spot, the first was normal, but the second one was that B dideoxy, right? Here. So in this way, you can get three types of fragments here. We call them as differential fragments. So three types of fragments here. Obviously, we will also be getting the full length fragments. What if this C is also normal? It will continue. Similarly, you will get two types of fragments here, three types of fragments here, again, two types of fragments here. If you have those differential fragments, you can use the gel electrophoresis to separate them. And finally, we can read that gel. So this position on the gel indicates their nucleotide number. So as many fragments, as many bases, as many fragments, we must need. We must have the same number of fragments um, equal to the number of bases we want to sequence. So here we want to sequence 10, a sequence that is made up of 10 bases. So we needed to have 10 fragments and 10 bands or 10 spots are shown on that gel thing. So this one is the first one. Obviously, this is this guy. Second, third, fourth, fifth, sixth, seventh, eighth, ninth, tenth. We line them up and then we look into from which tube they are coming, and then that's obviously the same thing. This one is, for example, C. This one is coming from this lane. So that's also a C here. This guy is a here. It is this guy. In real life, we don't have all those things because if we know that thing, why we are doing that? That is just for our elaboration or demonstration purposes. So that's a cartoon picture, right? So in real life, we have the gels and we scan them. Initially we used to rely on eyes, right? So you needed to rely on your visual, uh, you know, instincts, your eyes. But later on, people realize because going beyond, you know, sometimes you get 50 fragments or 50 bands because a base number of base pair as it increases, number of bands also increases on the gel. And gels were also having some issues because to prepare a gel, it takes time, and then to run a gel, it takes time, and then to interpret a gel, again, it takes more time. So gels were having a limitation. So you were not able to go beyond, for example, 100 bases, right? So then there was another gentleman, that was Professor Laurie Hood at Caltech, right? California Institute of Technology. He automated that process where what he did was he replaced the gels with capillaries, right? And then he, rather than coming up with those dark bands, black and white bands, right? Black bands, obviously, on those white or you can say opaque color gels. He replaced them with those fluorophores. So we had those same nucleotides, dideoxyribonucleotides, but they are fluorescently labeled, right? So we put some chemicals, we call them as fluorophores. So then these fragments are fluorescently labeled. They have distinct colors. So C has this color, it's pink, I believe, and that one is yellow, and that one is sky blue, and that one is green, or some people say parrot green, whatever, right? So they run through a capillary. I can give you that paper of Professor Larry Hood, and you can see that anatomy of that system and that drawings that are indicating how the system they actually created, right? So, but this is just a history lesson just to make the ground for NGS technologies because that automated Sanger sequencing is still there for limited genomes, for limited genes, sorry, or some specific regions of some genes. We call them as targeted sequencing. Or, to verify the results which you get from NGS for some of the genes, uh, the reviewers and the research papers, the journals, they ask for having that Sanger sequencing also. So Sanger sequencing is still considered as the gold standard, because NGS we do at huge scale, at a massive scale, so there is the probability of errors also, right? But Sanger is, it takes small data, it takes more time, so that is considered as more accurate. But obviously, that is good for parts of one gene or few genes. You cannot go for whole genome or for whole transcriptome. Obviously, that for that you need to rely on NGS or metagenomes, etc. Right. So here you connect them with a system. So they run in a capillary tube, and then they come to a place where you have an instrument, the sensor, or the camera that is recording, or basically in Sanger we get the peaks, the waves, we call them as chromatograms. We'll show you in a moment when we start processing that data, right? So and then in NGS something similar happens, but over there that base calling software is there, so it returns the bases as well as their qualities, right? So those fragments, they run into that capillary thing, and they are again running on the basis of their sizes. This is the smartest guy, second one, third one, fourth one, fifth one. So number one, position, and number two, color is important. You don't need to put them in four lanes, and you don't need to go zigzag like this, C, C, T, C, and then you look up and you say it's G. You get four colors, so you can quickly detect that this one is a T here, this one is a C here, this one is a G here, this one is a D here, this one is an A here, and so on. And in this way, you can get the longer length. So maximum lengths we were acquiring were around 1 kilo base, around 1,000 bases, right? And that Sanger machine, which we in fact had in our university also in good old days. Now it's not working. It's gone. To run that sequencer, the cost is more. For total reactions we can set up on that was 96. So it has 96 wells. So that it has a plate and that has wells. So over there you can place 96 fragments, 96 DNA fragments of length 1 kb. So maximum you can get from all 96 was 96 into 1, that is 96 kilobases, right? So that's a small as compared to NGS. So that used to take the whole genome, right, and sequence that in a couple of days, depending upon the genome size. So maximum it might take like a week, or minimum it can go for hours and sequence your data. So NGS or next generation sequencing is the second generation or next generation. Initially, when it started, they started calling it as next generation or NGS, but then they realized what we're gonna call this one. If this one is next, then this one should be previous. But as we mentioned yesterday, that people had a lot of respect for Fred Sanger. Obviously, that was kind of. Am I recording that? Yes, somebody pressed that. I think Sabia. So yeah, so people call that Sanger method as first generation and NGS as second generation. Then there is another good part of saying second was then we saw third also. That is the Oxford Nanopore, or PacBio, right? Or Helicose. So over there, there is one step which has been bypassed. We talked about that PCR thing, so that is not there. We'll cover that today also. So that is one thing, that is the good part which we had on those third generation sequencers. But obviously, still the NGS sequencer, the second generation, is famous and accurate and doing good. 3Gs are people are working on that. They are on trial basis. Some are working for smaller genomes, but these are the companies that are working in NGS Illumina that uses reversible terminators. In Sangers, those terminators are permanent, right? Those dideoxyribonucleotides. Once you remove that oxygen, you cannot put it back. Obviously, you can, but that has more cost of the reaction associated with it. So once to take out, that was not a good choice. So here they replace that with nitrogen molecule, not taking out oxygen, but they then replace hydrogen with nitrogen, right? And then when you need to reverse it, you just put water in it, and that hydrogen comes back, and nitrogen is removed, right? So in this way, you can reverse it. So that's called as reversible terminator sequencing. I will see in a moment. The company that did that was Illumina. Illumina, right? Maybe that illuminescence that happens when that light signal comes out when the nucleotide is incorporated. So that's why they named it as Illumina. Roche, the company that used to supply you the reagents and chemical, came up with pyrosequencing. So they captured that pyrophosphate molecule. It got famous as 454 because that was the number of the project. Thermo Fisher Scientific came up with what we call it as ion torrent. So they capture the ion, hydrogen ion that comes out from this. That's called as ion semiconductor sequencing. Applied Biosystem ABI came up with solid sequencing. Solid was sequencing by oligonucleotide insertion and detection, and that was, you know, it quickly lost the market. Initially, when it came out, that was very successful. But somehow they lost the pace, and obviously Illumina got the pace. Illumina is the, I would recommend that because that is, I believe, is the biggest amongst all, and that is giving us the better results. So next generation sequencing, NGS performs massively parallel sequencing, during which millions of fragments of DNA from a single sample are sequenced all together. So this is also called as high throughput sequencing. There is an input, you press a key to the computer input, the computer processes that in between, right? So like something is you take from your eyes, you see something that goes on your brain, and then you process that. So that processing thing is called as throughput. These are the high throughput. And what computer returns in return, or what you do, you react, you give a reaction. So that reaction is output, right? So what computer returns is output. So what's between input and output is what we call it as throughput, right? So you can process thousands or billions of sequences concurrently. So that's why these are high throughput. Generally, more than 500 sequencing by synthesis operations run in parallel, right? So in parallel, you process more than 500,000 sequences or fragments inside those machines. They are instruments, the sequencing instruments. They are really smart, so on which they process those maybe more than 500,000 reactions, right? In parallel. Okay, using state-of-the-art sequencing methods in efficient way. We should better call it as high-throughput sequencing, but NGS was the most famous or catchy word, so that's why it's out. So we are calling this online course as NGS. We did not call it as HDS, probably, right, to catch more of your attraction. So what happens over here is that DNA thing which you want to sequence on those machines is a big entity, right? So you need to break it into those small fragments, short fragments. And then there were the methods which people tried, like chemical degradation, the enzyme, the restriction cutters they used. But most famous was using the sound waves, the sonic emitters, right? The instruments, and then they cut those DNA fragments into small pieces. You fix the frequency of sound, and it is recommended if you want to cut it, that much should be the frequency. You just rotate a knob, and that instrument was there in ICCBS, and that was for this fragmentation thing. Okay, so we have it one of the finest sequencing center, Dr. Iqbal. He's attending that. One of our lovely fellow. He's part of ICCBS team, Dr. Ishtiaq, and his team is working there in ICCBS. ICCBS is the International Center for Chemical and Biological Sciences. So it has multiple centers. So one of them is Jamilur Rahman Center for Genome Research, right? So they had these instruments over there in our country, and people whoever having those sequencing facilities know they are around at multiple places in the country. And obviously, the people sitting in Middle East, you might have observed a sequencing facility nearby locations. Maybe you can go and check that instruments. Right, so somebody looked these fragments like fire of a shotgun. So this method is also got famous as shotgun method. And these are the instruments on which we sequence them. So Illumina came up with flow cell. Again, another a smart idea, a very efficient thing they designed. Obviously, their engineers, their R&D people were smart, right? So they came up with this instrument, flow cell. And one company comes with flow cell. So that flow cell has those lanes or channels in them. They're like your glass slides on which you do those experiments under the microscopes. You want to see something under a microscope on glass slides. So they are a little bit larger than that glass slide, these flow cells, right? I got a chance to see the flow cells. You can just Google and you can see the videos on YouTube. You can have it, right? So, yeah. So these instruments, they have channels in them, these flow cells. On those channels, there is a lawn of primer that is present on them. We are just seeing the cross section of these things. So on those lanes, these are the primers which are there. Those fragments they go in and attach to those primers, and first they replicate, they multiply, they do PCRs, right, and they get their multiple copies. Why? We talked about that yesterday, but some, I guess, quite a significant amount of the people are joining this course today. So let me refresh ones who already did that and the ones who are new. Let me cover that part one more time, right? So that these primers they attach those fragments here and then they do the sequencing over there. In solid, there was the flow chip that was similar to flow cell, but obviously you cannot exactly copy the same thing. That's their right, that's their patent. So they came up with those open channels, but that is something similar to this one. 454 came up with PTPs, picotitor plates, the plate-like things. Those plates have wells in them, right? Holes in them, just like the holes you see in what is that game, right? Yeah, golf, golf, right? So just like those holes which are here, and you put those balls into those holes. Obviously, there is a ball coming in. We'll just show you in a moment. So we do that sequencing on ball-like things. Those balls they are placed on those holes. So you're playing polo here. No, sorry, golf here. On torrent, right? So in on torrent, you do sequencing on those instruments called as OIN semiconductor chips, right? So remember we talked about hydrogen ion that comes out in this PTPs. We have that pyrophosphate capturing thing. This had a different algorithm, but that's obsolete. We are not covering that. This one we will just cover in a couple of slides in a comparative overview of those sequencing technologies. And right, what happens over here is that OIN torrent has those hydrogen ions, right? That are that are released when nucleotide is incorporated, and those hydrogen ions, what they do is they change the pH, right, of the medium. So those small ball-like things are added here. Again, they had the holes, and when you see a cross section, it's coming in the next slides. So over there, we have those something like those sensors. They are like small pH meters. So those sensors they detect the change in the pH. If nucleotide is incorporated, there will be a change in the pH. If not incorporated, no change, right? And that change is then detected, and then wherever those pH changes are happening over there, we believe that nucleotides has been incorporated, and we can check which nucleotides has been incorporated, right? And in this way, we can sequence them. So this is how this whole party is set up. We do experimentation and we extract DNA and RNA. That's what the biologists, mostly life scientists, animal scientists, veterinarians, the people who are working in molecular labs, they are working on that. And these library preps and those sort of things. So normally you gave them the samples as the materials which your sequencing facility guys ask, right? So like in one of our experiments, Alpha Genomics, a local company that does this commercial sequencing. So they asked us to give just the leaves. So we cut the leaves since we were working with plants. For the humans, we gave them the blood samples, right? And then we extracted the DNA and we gave them the DNA. For RNA, we just gave them the leaves. For the plant data, we were doing RNA-seq, right? For the metagenomic samples, you gave the samples as such, stool samples or body secretions or blood or even soil samples if you want to get a metagenome from soil, right? Some people working on oceans biodiversity just give them the soil from the from the base of an ocean at different depths, depends where they can penetrate, and just looking into the poops of the fishes and other animals, which are the sea animals. You can detect which animals are there. What is the biodiversity of your sea? You can get the biodiversity of your skies also, right? You can collect the birds, right? The poops. Some people are doing that. Very interesting projects, and in this way you can get the migratory patterns of the birds, or you can get the biodiversity, right? The people working on antimicrobial resistance are also doing that from poultry, similar things, right? So that is called as metagenomics, right? So that many things which you can do. So depending upon the data which you are processing and the requirement which you are given from your sequencing company, you do those library preps, please. Dr. Mudassir, class हो रही? Very good. Okay, in PCR, what you do is why we need to do PCR here. We covered that part. So here we believe that when a nucleotide is incorporated, a color signal comes out, right? And that light signal is hard to detect. Okay, if it is only one nucleotide incorporated into just one fragment. So what we can do is we can increase those fragments. We can copy them. We can do PCR for those fragments. So here, and this is shown in red, this third layer. The first layer is experimentation. The second layer is library preps. And this third layer is where we are doing PCR. So on one side is bridge amplification or PCR on slide. Here you do it, I mean, the same slide on which you are doing the sequencing. So that's the smart idea. But for the other companies, we need to have another instrument to do that emulsion PCR thing. So again, for that solid sequencer at ICCBS, they showed me an instrument to do that emulsion PCR. Again, that emulsion PCR is a little bit complicated, right? So that is the strength of Illumina over here, that it is winning on all of its protocols and methods. So then for sequencing, OIN Semiconductor came up with capturing that hydrogen ion. Pyrophosphate came up with capturing that pyrophosphate. Ligation by solid, right? And reversible terminators by Illumina. Again, Illumina wins here also, right? You must be asking me, maybe I'm a brand ambassador of Illumina. No, actually, my PhD data came out from Illumina, and throughout my career, my educational career, I have been working with Illumina. Probably Illumina has got the maximum market, and that was the first, technology that won the big market, especially in United States. So maybe because of that, but I think others, they also have their pros and cons, right? So you can always do a comparative analysis, and whatever suits you for your work, whatever machine is in your neighborhood. Again, for the read length or for the fragment length which they produce or sequence over there, fire sequencing had a strength because it was producing the longer reads. And then last layer is this data analysis. This course is all about that data analysis, right? So experimental designs can be anything like human subjects, plants, animals, mouse, bacteria on petri plates. If you are a data analyst, you would love the freedom of working and learning from people from different disciplines, right? The people coming from veterinary, animal sciences, the people coming from plant sciences, the people coming from. Do not restrict yourself to just one domain. If you are a data scientist or a data analyst, be flexible. Okay, don't say I'm just a microbiologist, so I'm not dealing with the data coming from plants. Or if I worked in during my PhD, I worked with plants. When I got back, people were offering me to work in microbiology on their metagenomic data. I would love to interact with them and learn from each other. So be flexible. Bioinformatics is a bridge between different disciplines, right? So you, we connect the people with each other. Library preps, mein, what you do is you take those fragments and you break them into those short fragments according to the specification of the sequencer. So if sequencer is saying you that give me the fragment of size 100, so you cut it at a size 100. Okay. If he's asking you to cut each fragment at a size of 150 basis, do it 150. If you give him 100, that is means you are underutilizing its resources. If you are giving him the length of 200, he's asking you 100. That extra 100 will be chopped off, right? It will not be sequenced. Okay, so sequencer will stop at 100 because that is its limitations. So beyond 100, it will start giving you error, or maybe it will not go further. Its reagents and chemicals or its instruments They will be exhausted. Right? So what you do over here is you prepare those fragments and then you attach what we call them as adapters. We talked about primers here. In as we said that there is a lot of primer that is embedded. You can ask me a question. There are millions, or there are like thousands of fragments. That's what we said. More than I guess half a million, 500,000 reactions are happening in parallel, right? So what should we design those half a million primers for all the fragments, like one for each? No, we cannot do that. So for that, they came up with what they call it as a universal priming concept. So we attach something which we already know, we call it as adapter, and then those primers are attached to them just to distinguish between the forward and reverse, or forward and reverse primers. We just color them green and red, but otherwise they are not naturally colored, right? They are different primers on both sides. I mean, one on for this side and one for that side. We know that we have two strands, and those two strands will be sequenced together. Both of them will become templates, and then we need to sequence them. So we need to have the primers for both of them, right? For RNA, we need to have convert that RNA to cDNA, complementary DNA, right? In the end, in the sequencing machine cannot process RNA because RNA is unstable, degradable. So it degrades real quick. While we were doing that RNA-seq experiment in USA during my PhD, my lab mate Brian, he need to cut those leaves from soybean plants and put them into liquid nitrogen and then remove, yeah, then take them to there was an to put it there immediately, and then RNA was frozen. And when he used to take him back and extract the RNA and then to convert the cDNA, so for that he need to do, there is another thing called as freeze drying. There was another instrument. Now we are lucky that we got the chemicals. One of the chemical, excuse me, which we get from alpha genomics was RNA later, right? So RNA samples, uh, be it the tissue samples or be it the blood samples or be it the leaves or stems or any other thing, the bacteria from your Petri plate, they are immediately immediately put into RNA later, right? And then RNA later takes care of everything and it keeps the RNA preserved till it reaches to the sequencing facility. Then what sequencing facility does? It takes that RNA and convert to cDNA, right? And then make that DNA library. So rest of the things are the same. So within the sequencer, everything is DNA. There is no RNA sequencing within those sequencers. It's DNA, either cDNA or direct DNA from the genomes. Okay. Um, that is where we were taking, or we were cutting each and everything. So that thing is called as single end libraries, where we were cutting each and taking each and every fragment. In paired end sequencing, are the libraries are called as paired end libraries. By the way, this library word came from Sanger sequencing. Okay, so over there, if you remember or if you are familiar with the Human Genome Project, so while we were doing that, obviously human genome is big, so we were collecting the samples from different regions, right? So what people did was they need to store that DNA into restriction, they do the restriction cutters and put them into the what we call them as the carriers, the cloning vectors, right? So, have you ever heard of cloning vectors? Anybody? Just give me some examples so that I can take a couple of more skips from my tips from my coffee. Cloning vectors.
like
Any idea?
EasyJet, PTZ,
कोई आसान सा बताएं.
PTZ,
Plasmids, orchids.
Plasmids.
So for the Human Genome Project, they came up with something like they tried plasmids, but that was of smaller size. So they came up with GACs, yeast artificial chromosomes, and BACs, bacterial artificial chromosomes. So they took the bacteria, most of their parts of packaging material is bacteria, but deep inside they can store the fragment even more than length of the bacterial genomes or yeast genomes. So they were the artificially prepared chromosomes, where then they were actually putting the fragments from the human genome. So they were doing that fragmentation two times. So that's why it was called as double barrel shotgun sequencing. So if you maybe we can share that paper, that human genome project paper. That's so big. It's got so many pages, but multiple things which we can learn from there is those libraries, those cloning vector libraries. They were prepared, or they were the the prepared to store the original DNA fragments for the Human Genome Project. So that word is actually coming from that. So, but there is no cloning vector library in NGS. But the sample preps are just they got famous as with the word library preps or library preparations. Okay, do not confuse with the cloning vectors or cloning vector libraries. You don't need them. You are not storing the material. You are getting the DNA, fragmenting, and then processing through your machine. Over there, those machines took like 10, 12 years. In total, it took like 15 years to complete that project. Okay. And the cost for sequencing was $1 a base, right? So that was straight away 3.2 billion for sequencing. And rest of the expenditures you can add up. And there were like so many organizations all over the world in that human genome project that collaborated with each other to do this different. I mean, that was a marvelous and wonderful effort that gave us this boom to our subject, bioinformatics, and then this NGS came right after this human genome project because we got funding, and the commercial companies, they started thinking, developing something smarter than the Sanger sequencer. Okay, but obviously we should give full credit to Sanger and Professor Larry Wood for automating this sequencing thing, and people like him, right? And people who develop those reversible terminators, and people who are working on templates, or who are working on so multiple, I mean, it was whole community efforts, right? So that has multiple pieces, roles which people played. So here in paired end sequencing, what you do is you attach another sort of adapter. For example, we want this a longer fragment. I mean, we want it to be sequenced from here to here. Suppose these are just 100,000, or maybe just 1,000 bases here. And if we just capture their ends, like this part from here, this part from here, rest of the part we can also detect it. What we have in between, we can detect that because we will have a sequence of 100 bases on this side and a sequence of 100 bases on this side. In between, you can cut or you can drop the length which you have in between, that 800 bases in between. You don't need to sequence. So that can save your resources. Earlier, if you are spending $1,000, now you can do it in 200 US dollars, right? So that is just one advantage of doing that, but actually that is not the objective. That main objective was to do genome assemblies, and this thing will be covered when we talk about genome assemblies, right? A couple of weeks from now. So you get that adapter here, and then this adapter is having those invert repeats, right? So they are complementary to each other. So this thing like gets that into circle. You have a longer thread and you want its end. So what you do is you can tie a knot here, so that knot is here, right? And then you can do that restriction digestion. You can cleave and cut that part. Only take this part that has the adapter. So that has your ends. That part is an one is, for example, this is the forward end and this is the other end, right? So in this way you can get so these things are then when you sequence them, and then after the sequencing you can separate them out. So one of them is your forward read and one of them is your reverse read, or you can have they are one of the pairs. So one of them is first of the pair, and the other one is second of the pair. Or you can call them as some people call it as P1, and some people call this one as P2. What is the distance in between which you actually discarded? That thing is called as an insert. Insert. Just write it somewhere. So that insert over here in this example, we had 1,000 bases. When we have 100 here and 100 here, and we remove the insert, that the insert size was 800. So that 800 was discarded. That is the insert. Is it a long insert? Yes, it is a long insert. In some of the cases, you might have short inserts like 200 bases or 3 to 400 bases, right? So sometimes you can have even the inserts of maybe in kilo bases, even further longer inserts. So long inserts, short inserts. Insert is the piece which you actually discarded, right? I mean, that term confuses. Basically, it is deleted, but insert means there is something in between these two that needs to be inserted. So that's why they kept the word insert, right? Now we are coming to that amplification thing. So what happens in Illumina is it got famous as bridge amplification or PCR on slide. That's what we also call it. So within that slide, within that flow cell, what happens is those fragments they go in. When those fragments they go in, obviously they have an adhesiveness with the glass slides, so they stick to the glass slides. Then those fragments they have the adapters on both sides, and on the floor of that glass slide, we had those primers. So this primer of blue color is actually complementary to that. What is that? Purple color. So that is the adapter on this side. So this guy is complementary to this guy. When it comes in, I mean, obviously that DNA does not stand still. It floats, it moves, it dances, and something happens like as soon as it comes closer to its partner, right? It gets locked here, and then it starts then replicating. So as you see, it keeps on growing, growing, growing. We see that one side is having attached here. I mean, this guy has this side attached, the new one. The old strand has this side attached and the other side is free. And then they change the temperature within that sequencer, right? And these fragments, they get free, and then they move around and find new partners. Obviously, the primers are there down there on the floor. There is a lot of primer. Both types of primers are there. The blue one is complementary to purple, and purple is complementary to blue. And then finally, they keep on moving, and those bridges are formed. We will share the videos with you. Sadia, why don't you post the videos on WhatsApp of this phenomena thing, this bridge amplification, the ones on the group, and obviously for rest of the technologies also. Also share that TED talk, right?
I
So
have a question.
yes, please, yes, please.
Sir, in pair and sequencing, do we discard all of the insert or we retain some part of it except for the
So
adapter region?
we discard them, right? So we
So, sir,
discard
how do we design primers against the adapters? Because we don't know the sequence of the region yet. Or do we know the sequence of the region
them.
for which we design primers?
We know the sequence of adapters, right? So we know the sequence of adapters. So we design primer against the adapters only. Period, right?
And they're
So we
not
need.
the part of the template.
Yes, they are not the actual part of the template. Later on, we talked about QC, quality control. So over there, those adapters needs to be removed. Okay. So what sequencing company do is they actually remove those adapters before giving the data to you.
Sir,
But
if
since you
we
are
remove all the part of the template and just keep the adapters for which we already know the sequence, and that's also not the part of the template. So, what is the sequence for then?
you're talking about the paired end one,
Yes, yes.
what's happening over here is suppose we want to, detect there is an RNA molecule, whether it's present or how many copies it has. So we get it's one copy, two, three, four. All of them are of suppose length 1 kilobase, 1000 bases, right? If we rather than taking that whole 1 KB or 1000 bases, if we just take 100 bases on this side and
Okay, so
100
we
bases,
do keep some part of it, right?
yeah, 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 yeah.
Okay, okay,
Not
thank
only
you.
the adapter, yeah, not only the adapter, but we also keep
Sir, I got it. Thank you.
certification of the sequences. Yeah, we are not only retaining the adapters here. Okay, so what you do over here is you just get the multiple copies. Acha, now these things are called as clusters. So these clusters are generated. So this guy got this cluster, this guy got this cluster. Each fragment is multiplied how many times? They say it should be somewhere between 10,000 to 20,000 copies each. Why we need that? Why we do that? Because when next, when each fragment is multiplied 10 to 20,000, all 10 to 20,000 copies incorporate that nucleotide all at once, all together. So if at one spot there is one nucleotide incorporated and that light signal comes out, it is very, very small. Since here we need to install the cameras to take the picture from the top. So those cameras need to be very, very, very high resolution. So obviously their cost will be then gone way beyond our limits. Okay. So they were using uh, not the ordinary, but little bit advanced digital cameras. So in order to bring those fragments or bring those light signals to the range of those cameras, what they decided was to amplify those fragments. So each fragment is amplified multiple times. Okay, up till 10 to 20,000 copies. And here is just for one elaboration. Now, once they are multiplied, now that sequencing primer comes in and attaches here, and the nucleotides they start incorporating one after another, and that light signal. And then this is happening to all 10, 20,000 copies of the same fragment. So they are generating the very bright light signal good for digital camera. What if you have 10,000 copies of one fragment and 5,000 of them are giving you blue color and rest 5,000 are giving you another color like green or something else? So that means there is a noise here. So obviously the quality of that signal is going down. What if 10,000 out of 10,000 are indicating just one color, one red color? So you will get a very, very sharp red color. That means the quality of that signal is very, very high. So just keep it in your mind, the quality also. So two things are important, what base is here and how strong and how good is your signal, right? The quality. What base is there is called as base calling, right? And the quality thing is called as quality scores or quality calibration, right? Anyways, in emulsion PCR, what you do is these fragments, you put them onto those bead-like things, right? The balls, the beads, and all three technologies agreed to adopt that emulsion PCR thing. And what you do next is you take oil and then you put water in it and then you shake it. So when you shake it, those water droplets they are formed here within that oil, right? And what we try to do over here is that there should be a single water droplet containing a single bead as well as a single fragment on it, right? So look how simple I am. What I am expecting. Am I not expecting too much here? Yes, right? So again, I repeat. So what we want is those fragments need to be in single stranded shape. They need to be attached on those beads. So what they do is they take the beads, they take the water quantity, they put it into oil, each and everything, right? And then they start shaking. So as many fragments, as many water droplets, as many beads should be there. If there are 1,000 fragments, we need 1,000 beads, and we want those 1,000 water droplets to be there, right? To incorporate that. Obviously, you maintain it with that instrument. So over there, it shakes depending upon how much is your data, how much is how many fragments you have. Okay. And finally, what happens over here in this aqueous medium, it's a PCR reaction. So each fragment is then multiplied here. So each fragment get its multiple copies, right? So finally, and this thing is called as monoclonal. So only one type of fragment is here. We get its multiple copies, and then we put it into those wells in case of Roche, right? But for in torrent,
Can I
we
ask
load
it?
them. Yes, please. Question. Okay. ये भी करते हैं इसका जो पेपर है मैथ्स कर जिसके अंदर ये टेक्नोलॉजी है सारी. Anyways, so we are sharing some material, videos, papers, and obviously you can see them. Them using the whole week, and next week we meet. Uh, maybe we can. Acha, so next, no, so we see that PCR thing. Which one was good? Which one was more interesting? Which one you found was more, more catchy? This one or this one? Emulsion or bridge amplification?
Sir,
This one you
bridge
need to do it on a, yeah. So this one you need. This one is the instrument is taking care of each and everything. Here, this one you need to do it on a separate instrument. And then there was a time when these technologies were having good market, that they were looking for expert to do that emulsion PCR more than the data analyst as well as the sequencing guy, right? So that emulsion PCR expert, they got short in the market, so they were paying very heavy. I mean, there was a time when they were looking for them, right? Till Illumina get most of the market. So Illumina came up with another wonderful thing here. They retained the concept of those dideoxyribonucleotides, but again, rather than going with dideoxy thing, they put N3 here. They did not suffocate that molecule, and they also incorporated that fluorophore thing. So that fluorophore thing was already there in Sanger. So they came up with reversible terminators. So replaced H by N3. What happens is nucleotide is incorporated, light signal is released, and four color light is released. So green means another nucleotide. There are four nucleotides, four different colors, right? Obviously they have a convention, same as Sanger, right? So green represents the same nucleotide throughout. That's consistent. Okay. Uh, we'll show their color scheme in a moment. I don't exactly remember. Green was what? Yeah. See that paper. So in that paper, there's, and I guess C is green and red was T, red was A, and so on. Anyways, so what happens over here is that fluorophore molecule is removed, right? And then you put water back to the medium. Obviously, water molecules are there, but that gives a time as soon as you put those nucleotides, they are incorporated. That fluorophore is removed, that light color is detected, and then this water is added, and N goes out, and H comes in, so it becomes OH again, and this thing keeps on going. So you do that in cycles. Better we can understand from here. So here your I is a camera in a sequencer. C is green, A is blue, T is red, G is yellow. That's our scheme was here. And those are the spots which we are looking. So that is the spot at first time interval. I mean, I'm not talking about the seconds or millisecond or microsecond. Obviously, this thing happens real quick, right? So at fragment, actually one fragment has multiple copies here. So all copies, they are incorporating a green color, right? At time point one. In time point two, they are incorporating the next nucleotide, right? So that one is A. This one is C. This one is again, sorry, this one is T. The green one is C. When you see yellow, that's C here. You see red, that's again a T here. So top one is this, and bottom one is all. I'm not bothering you much. They are all green, so they are all C's here, right? So top one is CATCET. I mean, here I'm just showing you the hexamer, the six of them, but actually in real life they can be 100 or 150 or 250 depending upon the specification of machine. So Illumina was going from 50 to 250, right? So I guess MySeq goes up till 250. Now they are going for longer reads also, because now the research papers, they are also asking to go for longer reads, especially for genome assembly projects, right? So that's why people were referring to 3G technology. So Illumina is coming for longer read technologies also, right? But right now, the 3G is where they are producing those longer read fragments. What is a read fragment, by the way? I'm sorry, I was using the word read. So that fragment, after it is being read by the machine, is called as a read fragment. That is abbreviated as read. Okay. So that's why this technology got famous as short read technology, a short read sequencing technology. The second generation are NGS. The third one, I mean, what was the length which Sanger was giving us? I mentioned before. So how long a fragment could be, which Sanger can sequence for you? Up till 1000 bases, kilo bases. But some people say that up till 700 is the best. So if you go beyond 700 or 750, yeah. That so, why we call the Lumina reversible terminator. Yeah, not get the concept. Yeah, reversible इसलिए this terminator does not remove oxygen here. If you remove it, it's hard to put it back. Other than that, they replace H with N3. So once the nucleotide is incorporated, that color signal is detected. We know what we have right now here. Then, then you add water back to it, and this hydrogen comes back, and that N3 goes with probably with uh, with OH, maybe NH4OH or something is formed in parallel, right? But your hydrogen is then it comes back, so you get this situation. So now next nucleotide can comes and attaches here. In dideoxy things, when those nucleotides were added, since you had taken that oxygen out, oxygen is more important than hydrogen, right? So here its hydrogen is replaced by N3 and then this thing is reversed. So that's why that OH thing is then now available for next nucleotide to comes in. But after that signal comes out, this thing stays here. So it's a terminator, but not a permanent terminator like dideoxy thing. It is a reversible. You can reverse that, reverse it. That's why these are reversible terminators. So something happens here. So you get the spots which are red in color. Red means which nucleotide has been incorporated there, right? Green means the other one, blue means the other one. And this happens real quick frame of time. And there is a camera up there who's taking the picture. He knows which spot is this. So for example, this one is your read fragment one. This is two, three, four, five, six. I mean, it numbers them based upon their indexes, and finally it returns you a text file, right? That has those millions of those read fragments labeled as R1, R2, R3, R4, right? In 454, what happens over here is we know that when a nucleotide is incorporated, what is that key thing here? That is the bead, and that has the multiple copies. And here they are just showing you just one fragment or one sequence or one copy of it, right? And there is that primer and there is that polymerase going in, and there is a nucleotide that needs to be incorporated. So on opposite strand, there is an A, that means a T should be added here. So when that T is added, that I mean, whenever a nucleotide is added, a pyrophosphate comes out. So that pyrophosphate is then allowed to convert with another molecule APS. With this pyrophosphate, it becomes ATP, and then there is a luciferin that also takes part in this, right? And sulfurylase and luciferase, some chemicals they are added here. Finally, with that ATP, they ignite a light, and that light signal comes out, right? So that pyrophosphate, it's having a energy, high energy, that is used to make an ATP, and then this ATP is used in this reaction, right? And finally, we get light plus oxyluciferin, right? So this way, uh, we get a light signal here, right? But this light is of only one color. Remember the bulbs we had in good old days of golden color light. Now we have LEDs of multiple colors, right? Our lights are more brighter. Okay, but good old days we only had bulbs. Okay, yeah. So here, what you do in the iron semiconductor is those iron semiconductor chips. You take that bead again, immersion PCR, and the bead with multiple copies of the same fragments, monoclonal amplification, right? Monoclonal means the fragment of one type only. If you get a fragment that has, if you get a bead where you get accidentally, you get like here, here, if you get two fragments here of different type, it's now no more monoclonal. So both of them will be multiplied. So half of them will be incorporating another base, half will be incorporating another. So there will be a mess here. So here you get the multiple copies of the same fragment monoclonally amplified, and all of them they are releasing the hydrogen ion. Obviously, there is a sensor, so that's detecting the change in pH. So if there is a change in the pH, what happens is that is an indication that the nucleotide has been incorporated. Okay, but again, the hydrogen ion chain is the same for A and T and B and C. Like we had the same color light for A, T, G, and C. Then how can you distinguish like whether the A is incorporated or a T is incorporated. Look here, they have four colored light signals. Here, it's only monochromatic, only one color. Here, it's the same change in the pH, no change in the strength of the pH. pH is coming from hydrogen ion. It has nothing to do with the nitrogenous base on those nucleotides. So they make sure this thing, or they ensure where that nucleotide is incorporated by sending them in cycles. So first they wash those wells of this thing, this what is that? The picotiter plate with A's, and then they take the A's back, and then they send the T's, and then they send the T's back, and then they send the G's, and so on. So we do that in cycles in both the cases, right? So we wash those wells with different nucleotides one at a time. So all those spots where that hydrogen ion change happens, or all those where that light signal is coming. Here we have the wells where that light signal is coming, um, or where that pH change is detected. That means the nucleotide has been incorporated over there, right? So, for example, here nucleotide comes in and matches, and hydrogen ion comes out. So that is the change in the pH. Here you are adding a nucleotide, but it's not matching, so there is no hydrogen ion change. So you cannot detect what you get. So there is no signal here, right? Again, you do that for the next one. So two bases, for example, added, there is a double change in the pH. So that means two nucleotides has been incorporated. Similar happens here. But again, then it generates an error. We call it as homopolymer error. What if there are three hydrogen ions, or four of them, or five of them? What if the runs of the same nucleotide comes in? So over there, those sequencers, they are giving us some errors. So they are called as homopolymer errors. So that is the downside of those machines. These are the machines, some of the representatives ones. My data actually came from HiSeq 2000, Illumina 5500 XL solid ABI. ICCBS had it, but I think now it got retired. GSFLX 454 from Roche was very famous for longer reads. So for short read or for RNA sake or other projects, people were preferring this one. For genome assemblies, it stayed there for a while. Iron Torrent, Iron PGM looks like this. We had it in the region also. I think KRL Hospital, they have it, and some of the other organizations in the country, they have it. I think in Lahore, Lab Genetics company was using Iron Torrent. And JS Junior is a small system like this MySeq that is present at multiple locations in our country. That is good for smaller genomes up to a size of 16 GB. Its throughput is 16 GB, 16 gigabases. Human genome has what size? What is the size of the human genome?
3 billion base pairs.
Yeah, 3 GB, 3.2 in fact. So 3 GB. So in 16 GB, how many expected copies of genome you can have it? Divided by 3. So maybe 5 copies. So that's called as coverage. But for a genome assembly project, you need to have at least 30 copies of the same genome. That is the minimum recommended. It's called as coverage, 30x. For smaller genomes, bacteria, viruses, for metagenomic studies, this one is good. We have a one video of that metagenomics also shared real quick. So, right, so that's the MySeq, but obviously over there at that time its cost was 150,000 US dollars. Um, at that time, I guess when my PhD was finishing, this one was around, I guess, 6,000 to 7,000. Uh, no, sorry, 6 to कितना होगा ये 150,000, so 650,000, right? 650 or 750,000 US dollars at that time. This thing in ICCBS, they got it for, I guess, half a price, 150,000 US dollars at that time, right? So anyways, those machines are expensive. And the most important thing is not to buy them, but to run them. So once you acquire them, you need to keep them alive, to keep them running. For that, you need to bear the cost of reagents and chemicals. So if you don't have a sequencing facility near around, don't try to set up a sequencing facility. Try to collaborate with the ones which are already around you. This way, it will be good for both of you. But the commercial companies sometimes they provide us, since they do that at commercial basis at a huge scale, so we can get the good quality data on a cheaper rates, right? Next generation sequencing.
Excuse
So
me,
there is
sir.
NGS. Yes, please.
Sir, I have a question regarding Illumina bridge sequencing. You showed the signals of different color, and you said that we rely on the quality based on the number of fragments, giving the same color signal, right? So, what if that
Yeah.
region is heterozygous, that particular nucleotide? So, won't we get two color signals there?
Obviously, you will be having it's within the same. When you talk about those variant callings, that when we reach there on exomes or over there, we will be considering that. So at the time, you will have just one fragment. Okay. So at multiple locations, since we are doing this at this fragmentation at random, right? So, so we are taking individuals, the both copies of the genes separately, right? So while you do that library preps, they are separate from each other, so it won't be having any issues. So it's not the I'm referring to the same fragment when it goes within the sequencer. Over there, it should not be duplicated within the sequencer at that PCR step. So if that is duplicated, it's a PCR duplicate. If that duplicate is there by nature, obviously we had the duplicates, we had the uh, you know um, homologous regions within the original genomes and the DNAs, right? Multiple copies which are present. So there is a method over there that can detect between the PCR duplicates and the true duplicates. So here I was referring only to the PCR duplicates. So when once you cut them, when once you process them, we just want to make sure that this fragment should be just single here and here in that bridge amplification thing. When they become two, they should get separate immediately from each other, right? And start developing their own clusters. Again, at any stage, if there is twinning, if they stay nearby to each other, so they will be treated or taken as one cluster. So over there, that duplication thing is not that good. Okay, but actual or real duplicates do, yeah, they do exist, and we don't have any problems dealing with them. When we talk about data analysis, there is a method over there where you remove those PCR duplicates. So I was referring to those PCR duplicates here. I hope that helps.
Yes, sir. What I get that we sequence a single fragment or single template of DNA, it will only have one pair base.
Yeah,
So it will give the same signal. So it also
yeah.
means that we will have multiple copies of the same fragment, right? On the
Uh,
bridge.
in the end, each cluster is actually coming from the same fragment. So this one was not only one. That spot was made up of thousands, 10 or 20,000 copies of the same fragment, and all of them they incorporated a green color here. So I'm saying like maybe at some stage we got those twins, like monozygotic twins, so they could not separate at two fragment stage, but both stayed them together. So some of them were given us that green color, and some of them was given us, for example, blue color. We might be having a purple color in between. So that purple color means that the quality of that base calling thing is not that good, right? So system gets confused between whether it's a A or a T here, right? So that thing is not good. That's what I was mentioning. Okay, right? So next generation sequencing. अब next से next भी आ गई है. So where you gonna stay? You called it as NGS. Now what about this one? So better we should go with 1GS, 2GS, 3GS. Probably they have done the R&D for fourth or fifth GS, which they have not marketed yet, which they have not yet launched. But right now these things are coming to the market. So here, so this PCR thing which we were doing because of capturing that picture, so we were multiplying those sequences and we were getting their multiple copies. That step you don't need it here. So what you do over here is, for example, this Oxford Nanocore. I'm just mentioning only one of them. There is Helicose and there is PacBio. We can also share their material and just go through them, but just you know here and store your these names into your memory. Helicose and Pacific Biosystem, PacBio. These are other sequencers coming from this third generation NGS system. Yeah. So here, what we do is in Oxford Nanocore, they come up with a system of membranes, right? So here, you what you do is you take that template in a single stranded. You don't need to cut into small pieces, and that uh, proteins which are put on those membranes, just like the proteins which are present on your cell membrane, the transporter proteins. So they try to make those proteins like them, and that proteins has a small hole in them, a nanometer size. That's why it's called as nanopore sequencing, right? If you look into the width of your DNA, that's in nanometers, okay, or maybe even smaller than that, angstroms. So that should be where it can come and follow those nucleotides. They should pass on through this, and there is some movement that is going on here. I mean, some changes, physiological changes are happening, right? So what happens over here is this one protein unzips DNA helix into two strands. The second one then creates a pore in the membrane and holds an adapter molecule, right? That adapter molecule keeps bases in place long enough for them to be identified electronically. So there is a molecule here. There is some electrical system going on. So what happens over here? A flow of ions through the pore creates a current. Each base blocks flow of to a different degree. So you allow some ions to move. So as those ions they move, they strike on those nucleotides, right? And that generates some electrical signal. So that electrical signal is then adapter keeps track of them, and then that signal is connected to some system, and that change in that electrical signal is measured accordingly. So A change is different than T, then G, then C. So this way you can detect them. It's just like you are sitting on your window and some vehicles are passing, and then you are detecting that which type of vehicle passed. Then you can do that. It's a car, it's a truck, a motorcycle, or something like that. So what's happening over here is you don't need to do PCR. Why you don't need to do PCR? Because that's the real-time sequencing. You're not taking the picture. It's not sequencing by senses. That's why it's called as real-time sequencing, right? So yeah. So library preps and sequencing. No PCR. No uh, issue of getting those PCR duplicates, because those PCR duplicates will give us the wrong variant calls. When we talk about SNPs or variants over there, they will create the problems for us. We don't like them. Okay. All right. So that is one of the example. Minivine, the small nanopore sequencer, a USB like, a little bit bigger than that. We had one group of professors from a US university last week, Minnesota. So they brought this sequencer and they were doing the mitochondria of some organisms. So one of the colleagues, he was working on ticks. So they were doing the sequencing of ticks on them, and it took like how much time? I guess on our system they took like, yeah, and they generated how much data? So that longer reads are produced like in KBs. So that is the good one. Yeah. So far what we have covered, generation of sequencing technologies, first gen Sanger sequencing, second or next is 454 Illumina, Solid, or Torrent, and third is this Helicose PacBio Oxford Nanopore. We should better call it high throughput sequencing rather than NGS. We should call it as HDS. But obviously, NGS is more famous. It's a fancy tag as compared to HDS. It's just a comparison. Sanger was producing the read length or fragment length 400 to 900 bases minimum. 400 maximum is 900, but 700 in between is a good number that recommended. On torrent can go up to 200 bases. Pyrosequencing was claiming to go up till 700. So I said as it was famous in assembly projects, so people were preferring longer reads. But obviously, since this thing came out that claims to go in kilobases, KBs, that's more good. But obviously, this thing is for smaller genomes. For larger ones, people are mixing both Illumina reads with PacBio reads. That's making a good combination. But I heard that Illumina is coming with those long read technologies real quick. So they probably would announce it soon. Sequencing by senses in Illumina, that's the method. IronTorrent is the method. Pyrosequencing is the method in 454 or Roche 454, and sequencing by ligation in solid. So they were producing 50 to 250 bases. They were producing 50 to 85 bases, right? Accuracy is 99.9, 98%, 99.9, 98. I mean, everyone claims they are accurate. Reads per run, it's not applicable here. 96 well plate means 96 reactions, depends upon how big is your plate, how many wells it has. So that's not important here, but they can go up till 5 million. 5 million of size 200. That is what? 200 into 5 million. That makes 1 billion. So 1 GB data, you can produce it, right? Up to 5 million reads. I'm not talking about 5 million base pairs. Each read or each fragment of size 200, you can multiply that. So 700 into 1 million, that's 700 million. 1 million reads are produced per run. That was given from one of their largest machines at that time, right? So that slide may be a little bit old. So now that statistics might have been changed because Illumina came up with high scan after high seek. So high seek was up till 3 billion. 3 billion of size 100. So high seek. My seek was up till 250. Okay, 150 to 250 was recommended on my seek. iSeek was up till 100 base pairs or 100 bases. This one was 1.2 to 1.4 billion. Time per run 20 minutes to 3 hours for one run. All 96 uh, well wells in a plate, right? So our 96 KB of data to produce 2 hours here, 4 hours here. This one for 1 to 10 days, depending upon the sequencer. So obviously 10 days for high seek at full capacity, maybe 1 to 2 weeks. Cost per 1 million basis, $2,400. So if your data is big, cost is heavy, so we won't recommend $1, $10, 5 cents to 15 cents, 13 cents. Advantage is longer individual reads, useful for many applications. Smaller genomes, you want to verify your NGS results, feel free to do that, especially the people who are working with exomes, who are working with, human genetics or genomics, right? The exomes and genomes where you're looking for SNPs, right? So there you would probably verify your results with this because that is going at huge or massive scale. So it's not bothering much the individual genes, but some of the genes may be really important for your research since most of us or all of us are classical researchers. We are familiar with working with one or two or three genes, but not with the huge amount of genes. So people are still, fitting those classical genetics models here. So that's why they recommend doing this thing also. Long read size over here. So less expensive, fast here, longer reads. The potential for high sequencing depending upon the sequencer model, low cost per base. Disadvantage is this one is more expensive. Homopolymer errors. Runs are expensive. Again, homopolymer. What were the homopolymers? We covered that. Anyone?
The same nucleotide when it is repeated
Uh,
again and again.
so equipment can be very expensive and slower than others, right? So yeah. Next is applications. But before we talk about the applications, I have a request. Why don't you do this part because we are short of time and I was thinking the things. So this thing is, for example, some of the databases where you can get that data for the individuals' genomes, where we had those model organisms like Haemophilus influenzae. We had its own database, right? And then this C. elegans, ASDB. The C. elegans database was the first one genome database to be developed. So you just click here, you just go in this slideshow mode, and when you click, that database will be opened on your system, and you just visit that just uh, to see how they manage those databases for those individual genomes. Right, for Arabidopsis, maybe thing on the other screen, show you when it pops up. So we have for tier 10 Arabidopsis information resource tier. The latest version was 10. I guess now the 11 is launched or not, but what I worked was EIR 10. That is for Arabidopsis. Saccharomyces yeast is this one. So that also is similar to this ASTB. My system is okay. No issues. No issues. Don't mind. That's ASTB. This is how it looks like. It also has the other model organisms, right? But for the humans, yeah, that's the Human Genome Project, National Human Genome Research Initiative, NHGRI, a federally funded organization, started in 1988. Francis Collins. Then Celra, a commercial company, also joined in 1998. They also, announced and both simultaneously announced the completion of the project in year 2000. 3.4 billion bases sequenced at a cost of $1 a base, right? Okay, 3.2 like right? 3.2 3.4. Is that right? 3.2, you know? Human genetics are there. 3.2. Uh, so that typo here. I don't know. I'll correct that later. I'll do that correction in your slides and in your mind. Right. So this one is for the human genome. They placed it on UCSC genome, UCSC University of California, Santa Cruz. Just click here, then it will show you different genomes which are published, which are placed there. Along with human, there are others also. So if you want to get their sequence data, the reference, you can get it from here. UCSC has genome browsers. We'll cover that when we talk about the uh, that exome thing or the resequencing thing. So there we will come back to this thing, but you can just see those, click and see those databases. Gene expression omnibus is having the gene expression data. Basically, it started with microarrays, but later on, everything is there. So we had the microarray and RNA sequencing. Again, these figures are old, so if you click, you can see that statistics to be updated, right? So GEO has those sort of data sets. We'll visit them later on when we will be doing RNA-seq. Just to give you an idea, these are the databases where you can get that. And GS data, you don't need to reproduce the data by yourself. So that is the expression profiling by high throughput sequencing. That is the RNA sequencing data, expression profiling by high throughput sequencing. Then we have the genome data, we have the microarray data, we have the SNP arrays, PCRs, right? Everything is here on GEO. So you can get that from these databases where you can have that data. If you know that data analysis thing, what happens over there is that people, they work on one or two aspects during their projects, but it is a requirement of the journals, the publication agencies, that you should publish your data to or store your data to GEO or some public repositories and share the link with the communities. So that huge amount of data sets are already there, right? So over there, people are taking, for example, our data sets like Pakistani. There is the one called as the thousand genome. I think that link I missed. Also mentioned that thousand genome has even your Pakistani genomes also. You can get them and you can work on them. Yeah, those links are already there. So that was EU. You see here in this paper, you get the link of that data set, so you can get access to data. You can download that data and then you can process with the techniques which we learn from that course. Even some of the students, we don't have those sequencing facilities. expensive or difficult, but you can get the online data sets. Even people working abroad, some students are working on those already published data sets during their doctorate in foreign countries also and developed countries also. There is no harm in it because normally it happens is like while I was working on soybean, we were only interested in its flowering gene networks. The rest of the things we were not discussing, like the yield or like the its height or its vegetative traits. Somebody else can take that. Obviously, that information is there. Everything is sequenced. It's there on Phytosome. It is the biggest database that has the plant data sets. ENCODE has this 1000 genome project. Initially, the effort was to collect 1000 genomes all across the world. Now they reached to 100,000 genomes, 1 lakh. And then now they reach to, I guess, 1 million. So now, right now it's like 1 million genomes. Okay, you can click that and see. So over there, they are checking, they are getting the genomes all across the world. I guess the Pakistani genomes over there are more than 200. So you can get them and you can, study uh, the population genomics. You can narrow down to some of the genes in which you are interested in, and you can design your query. I'm so fun on other screens. Yeah. So the International Genome Sample Resource supporting on human variation data. Now they have changed the names. Originally it was the 1000 Genome Project, right? And now it got uh, converted to International Genome Sample Resource, right? So they had the samples from all across the world. So people with their supercomputers are analyzing your data and my data or our population data, and that information is very important to developing or forecasting what diseases can be coming into the different regions of the world and how can we fight them, how can we make the medicines for them. That information can be very interesting for the pharma companies, right? That's what the way those things are going. And code another one, encyclopedia of DNA elements. So they are putting those promoters or other sequences, the patterns which we have in the genomes. So which patterns are rich in which, uh, you know, you know, populations, right? And SNPs and many other things, right? An exome sequencing project, NHLBI, National Heart, Lung, and Blood. So they are getting the samples from heart, lungs, and blood diseases mostly, or from these organs, and collecting the exomes of the patients having those diseases. That information. Autism, it has also. Human microbiome project was very interesting. So they wanted to collect the microbial samples. One of the project was very famous, American Gut Project, right? So you are getting the microbiome. People who are working in microbiology or metagenomics, they believe that we are more microbe or we are more bacteria than humans because those bacteria present in our gut, they are very important. If your gut is good, you are good. Okay, everything depends on your your gut, right? So obviously those bacteria, they drive our lives, they drive our moods. Okay, and again those bacteria are again influenced or changed by our habits, our dietary habits, how much water we drink, we do exercise or not, we live happy or sad, right? So all those things. And then this data analysis thing we start by next week. In between, maybe if you are interested or if you get a chance to get that, do we need that installations this week? Linux for okay. I think we should give you the time to settle down and to assimilate, especially those people who are new. Maybe you can go through these notes and these theory lectures, but I think I want this course to be more fun with the hands-on stuff. We want it to start early as possible. So that's between you and Sadia. Maybe you can start as you like. All right. So I guess that's enough. Pretty close to an end. Any questions, queries, concerns, feel free to ask, post on the chat box. Let me see if we have any questions there in the chat box. Got it. Thanks. Nothing else. Okay. Thank you so much. So, anything else? Comments, questions, concerns? Not.
Sir,
I would let you go,
sir,
guys. Yeah, man.
regarding the sir, you talked about something monoclonal in the emulsion PCR.
No.
I couldn't
Mhm.
understand that thing. What was that?
See,
You said there is
monoclonal
fragment.
means, clones of the same thing, right? So, original fragment only of one type, so that when you multiply, it should be just copies of only a single fragment. It should not be like two fragments they get in here, right? So, it should be just one fragment, okay? And all copies belonging to that. If two fragments they enter here, obviously some of them will be coming from fragment A, some of them will be coming from fragment B, half of them maybe, right? Depending on which stage the other fragment gets into it. Okay. Obviously, that will confuse the sequences. So it will be given like one of the fragments will be taking, its all copies will be taking A, and the others will be taking T's. So depends the strength of the signal, or that light signal. Over here, the light is of only one color. So, but obviously the strength of that light, obviously the light color is important and its intensity is important. So that will be low. So the quality will be low. So that thing is then converted into those quality scores. Here you get different colors. So like if half of them, for example, we look into its clusters. So there is a cluster. So ideally, it should be of only one type, right? So there is that. So if you have multiple types of fragments here, so obviously you will get multiple colors. So your quality will be decreased. So you'll not be getting that sharp green color. You might be getting some color like in between green and blue. Maybe what is that? What is the combination if you mix blue and green? Something like purple? No, it's sky blue. Sky blue. That's what she believes. What if you mix green and red? So in between them, somewhere around yellow? Yeah. Yeah. Okay, so that creates an issue. Anything else? No, Ji? Let me
Hello,
go.
sir. Hello.
Yes, please. Yes, please.
Ji, sir. My question is usually when we
Uh,
do
can
sequencing,
you can you speak a little bit louder?
okay, is it right now?
Yeah, yeah, please.
Okay, so usually whenever we do sequencing, the sequencing company tells us that we provide you 5 GB data or 10 GB data. My question is how
Oh,
they
yeah.
actually calculate this.
That thing we will cover. It's called as coverage. So, for example, your human genome is, let's suppose, round me up to 3 GBs, right? So, one copy is 3 GB. One base takes 1 byte on computer, right? So, 3 GB, whether you say it gigabases or gigabytes, it's the same thing. In order to get, for example, 30 copies of the human genome, you must have how many? 30 multiplied by 3. So your total data should be 90 GB, right? So if you
Okay,
generate a 90 GB data, so you divide that 90 with your genome size. So that will give you an idea of your coverage. So that is 30x.
okay, okay,
Okay.
okay.
So we'll cover
Excellent.
that.
Thank you.
So I think you deal with viruses or bacteria. So for example, if your genome is 1 MB and they are giving you suppose 1 GB. So 1 GB divided by 1 MB is how much? I guess that will be 1,000 copies of that thing, right? So that's how you do that. Okay. So how
Okay,
many bases you get in total? That is like if you are given, there's a formula with it. It has n times L. N is the number of reads you are getting multiplied by length. So length of each individual read fragment, whether it's 100 or 150, you multiply these two divided by genome size. Okay. So total bases you get divided by your genome size. That will give you an idea of the coverage. Anything else? recording. Stop recording. Okay, ji, thank you so much. See you next week. If you have any questions, concerns, queries, you may